Okay, welcome to lecture 4, and today we're going to be talking about skeletal muscle. And in this lecture, we are going to be applying some of the principles from Anatomy and Physiology 1 in terms of muscle structure in order for us to understand its function. So, to kind of review some of the major components, and again, for those of you who took Anatomy and Physiology 1, this should be kind of a review of some of the terms that are important for us to understand in terms of relating these components, like we mentioned, to the function of muscle. So we have the epimysium, perimysium, endomysium. Each of these three layers will be a connective tissue covering that are going to cover various components within the muscle. A fascicle is a bundle of muscle fibers. Muscle fiber, as most of you know, is a word that can be used interchangeably for muscle cell. So if you hear the word muscle fiber, it means the same thing as muscle cell. Myofibril are the protein elements that are going to make up the muscle itself. We have various types of myofibrils, and they will play an important role in the actual contraction process of the muscle itself. So on the next slide, we've got some images to kind of go along with these terms and to kind of help us to review these terms. So let's start with kind of the largest, most external layer here, the epimysium. Epi means upon. The epimysium is going to lie upon or around the entire muscle. Again, this is made up of a connective tissue, sometimes called a deep fascia. The epimysium will converge together at the end of the muscle and form the tissue that makes up the tendons. So that's your epimysium. Next we have perimysium. And perimysium is a connective tissue that surrounds each fascicle. So peri means around. This is a connective tissue layer that surrounds the muscle fascicles. And a muscle fascicle, as you can see here, is a bundle of muscle fibers. And the last layer of connective tissue that we want to look at is the endomysium. And the endomysium surrounds each individual muscle cell. And the endomysium, again, endo means within. It's a connective tissue within the muscle that surrounds each individual muscle cell or muscle fiber, and also synonymous with the cell membrane itself. Let's see, what else can we see in this image? So we can see a single muscle fiber. Again, each of these individual little circles represents one single muscle fiber or one single muscle cell. Within those, we have myofibrils of varying types. There are two major types, which we'll get into here in a little bit. But those are the protein filaments that are going to allow us to have the actual contraction of the muscle. So if we were to kind of take a microscope and zoom in on one single muscle fiber, there are various components that will be important for us in terms of playing a role in the actual sequence of events for muscle contraction. The sarcoplasm, again, sarcoplasm sounds similar to cytoplasm. Sarco refers to muscle, but the sarcoplasm is basically the cytoplasm within a muscle. Transverse tubules, sometimes called T-tubules, are basically an evagination of the cell membrane. So if this is your muscle cell, a T-tubule is like a dip, this little dip thing that I'm drawing, down into the muscle cell. There's not actually a hole, there's not actually a path here. It's just a ditch or an evagination within the cell membrane that's going to allow us to send an excitatory signal relatively quickly to the inside of the muscle cell. Sarcoplasmic reticulum sounds similar to endoplasmic reticulum, and it is very much so. The sarcoplasmic reticulum is specific to muscle, to skeletal muscle, and this is the endoplasmic reticulum within the skeletal muscle. and this serves as a calcium storage bin for the muscle cell itself. And that will play an important role for us when we talk about the sequence of events for muscle contraction. But basically, we have to have these calcium storage bins in order for us to keep the calcium in this hidden place when we don't want muscle contraction to occur. And again, as we've already defined, the myofibrils are the protein filaments that are involved in the contraction sequence. Okay, here you can kind of see a better image than what I could draw for you, but let's have a look-see here. You can see the T tubules. Let me see what color marker I can get here. So here you can see the T tubules. Again, it's just an invagination in the cell membrane. It's not actually a path. And the reason that we have these is it allows us to quickly send a signal to the middle of the muscle cell. Again, muscle cells can be relatively large depending on your on your training status and what types of activity you do. So it would be very important for us to be able to reach the middle in a timely fashion if we want to have some sort of quick motion or quick response to a given stimuli. So that is the role of these T tubules. Again, the sarcoplasmic reticulum is going to serve as a calcium storage bin for us, and that's actually located very close to the T tubules themselves. And together, the sarcoplasmic reticulum and the T tubules create what's called the triad. Again, tri means 3, and a triad is made up of both sides of the sarcoplasmic reticulum. So you have some over here, you have some here, and then also the T tubule that is in the middle. And this triad will be important for very efficient signaling of the muscle and also efficient release of calcium from the sarcoplasmic reticulum as well. And lastly, you can see the myofibrils up here. Again, these are the protein strands that are going to have an interaction in order for contraction to occur. Okay. As I have alluded, these myofibrils are the contractile elements of the skeletal muscle. They are long strands that are subdivided into smaller units known as sarcomeres. A sarcomere is considered the functional unit of contraction within the muscle. So in order for a muscle to shorten or to generate force or to have contraction, these sarcomeres must become shortened or have the myofibrils within the sarcomere kind of overlap, and that would allow the sarcomere to actually shorten. Okay, so here you can see one single muscle fiber. If we zoomed in on one single muscle fiber, and you can see all of the myofibrils that actually make up the muscle fiber itself. Again, the more highly trained you would be, the more myofibrils you would have within a given muscle cell. So, that's something that can definitely be variant depending on the person. But these myofibrils are directly related or directly linked to force generation. Okay. So in terms of unique characteristics of the skeletal muscle, you can see here that the skeletal muscle is also called striated in appearance, and that's because it's striped in nature. This striped appearance can be seen in skeletal muscle due to the organization and overlap of both actin and myosin. So we have actin, which is a thin filament, and myosin, which is our thick filament. And together, as they overlap, this creates some striations. And we've got some images that will kind of help to better explain that here in a second. Again, a sarcomere is our functional unit of contraction within the muscle, and it's made up of both thin and thick filaments. Okay, here you can see a histological view of skeletal muscle. So skeletal muscles are cylinder-like in nature. So again, you can see here it's kind of set up like a cylinder, basically. Striped in nature, so again, you can see those striations, and that has to do with the overlap of actin and myosin, creating these dark and light stripes. It's also multinucleated, and that basically means that for each individual cell, there are more than one nuclei. You can see that the nuclei kind of lie on the outside edge of the muscle cell. Other things to make mention, everything, well, not everything. Skeletal muscle is very organized in nature, particularly when we compare it to cardiac muscle, which is very branched in nature. Skeletal muscle is set up in a way in which it can contract in a shortening and very efficient way. So when skeletal muscle contracts, it shortens as a whole group, and that shortening motion allows us to actually have joint movement and generate force in order for us to have that occur. Okay, so here we can see what an actual sarcomere looks like. So again, you can see down here these blue lines are our thin filaments. The red lines are our thick filaments, also known as myosin. And, when muscle contraction occurs, we're going to take both ends of the sarcomere and they're going to slide together in order for us to shorten the sarcomere. I'm very excited that I just figured out that I can use my little pen marker deal on the slides as I record. So this is like a new discovery for me, so I'm sure I will completely overuse it in the next couple online lectures for you. So get excited for all of the marker that I'm going to be using on all of these. So anyway, I don't care if you know about the I band, the H band, blah, 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 all those names of those components within the sarcomere, but I do want you to understand the sarcomere is the functional unit of muscle contraction, and when a muscle shortens, the Z lines here are going to slide closer together. And not anything in particular actually shortens, we're just overlapping those filaments and kind of butting them up against each other. So the thin filaments are going to slide towards the center of the sarcomere, and them sliding towards the center of the sarcomere is what actually causes the sarcomere to shorten. Okay. So again, we've alluded to this idea of a thick filament and also a thin filament. Our thick filament is made of myosin, and myosin is a thick protein strand that has globular heads that protrude out. And these globular heads form what's called a cross bridge. And our cross bridges are what actually allow us to generate force. So the more cross bridges you have connecting to the actin molecule, the greater the force generation. Our thin filaments are made primarily of a protein called actin, and this is actually a much thinner molecule, of course, in comparison to the thick filament. It also contains two other protein particles known as troponin and tropomyosin. Okay, so here you can see another image of what a sarcomere looks like. In the green here, we can see the actin molecules. Let me get a good marker. Okay, so your actin is here, your thin strand. Again, we have troponin and tropomyosin. Tropomyosin is this black strand that kind of covers the actin molecule, and the troponin is this little yellow dot. And that little yellow dot will serve as a place for calcium to bind. It's a binding site for calcium. Okay. The myosin, thick filament, is this whole situation in the middle. And you can see these little yellow protrusions coming off of the myosin. Those are your myosin heads. Then the more myosin heads you have, the greater capacity you have to generate more force. So basically what happens when contraction occurs is these little myosin heads are going to bind on to actin and pull actin towards the middle, towards the center of the sarcomere, and that's what causes the shortening motion. Okay. Again, just zooming in to have a look at just one individual muscle protein interaction. So here you can see the actin molecules in purple, and those are made of basically two strands of beads kind of swiveled together. We have troponin here in yellow again, and troponin serves as a binding site for calcium. And when calcium binds to troponin, that causes the tropomyosin, this little pink strand, to move out of the way and uncover a binding site. Once that binding site has been uncovered, the myosin head will bind to that actin binding site, and that's what creates muscle contraction. In order for this to occur, we have to have ATP available, and as we bind to the myosin and actin together, that costs us energy, so we're going to break down the ATP into ADP and phosphate. Get a marker over here. So ATP must be present, and it will be broken down into ADP and phosphate each time we have a binding of the myosin to the actin molecule. All right, so energy must be available in the form of ATP. We also must have calcium available to uncover the actin binding sites. Okay, so here are some terms that we would like to define before we move forward with the sequence of events associated with muscle contraction. So to start, we'll start with the alpha motor neuron. And if you remember to the nervous system from Anatomy 1, we talked about how a motor neuron would send a signal or carry instructions in order for us to excite or stimulate an effect. Okay, so a motor neuron is going to cause the contraction of the muscle, essentially. An alpha motor neuron, if you think about alpha males, like in a dog pack or something, the alpha is the one who is in charge or the most dominant. So the alpha motor neuron is in charge of this motor activity or in charge of stimulating this motor activity. A motor unit is an alpha motor neuron and all the muscle cells that he is in charge of. Okay, so you could have a small motor unit, it could be one neuron and maybe like 7 muscle fibers. Okay, you could have a large motor unit, which would involve one alpha motor neuron and like 400 muscle cells or something. So the size of the motor unit is determined by the number of muscle cells involved, although for each motor unit, there's only one alpha motor neuron. So you can think of this like from a business perspective, a motor unit is like one factory, okay? So a factory could be very small, so you have one boss who is your alpha motor neuron, and maybe like 7 workers, okay? So this factory is like pretty small. They're making something on a small scale. Or maybe your factory is really, really big. Maybe we're looking at, one boss, but then maybe you have like 400 workers in this given factory. All right. So again, with either type of factory, you still only have one boss, but the number of workers can vary from factory to factory. Okay. The people that work in your factories can only work in one place. All right. So one single muscle fiber can only be a part of one single motor unit. All right. These are exclusive, okay? So there's no way for a neuron to be in more than one factory, and there's no way for a single muscle cell to be in more than one factory, okay? So they're very exclusive in nature. The neuromuscular junction is exactly what it sounds like. It's the relationship with the alpha motor neuron and the muscle cell itself. These are going to form a junction. Do keep in mind that they don't physically make any type of contact with each other, but they are going to, this is a place where we can relay the signal from the neuron to the muscle cell. Okay, so in terms of the events involved in muscle fiber contraction, this is a fairly complex set of reactions that will occur in order for a muscle fiber to contract. So first thing that happens, an action potential is sent from the alpha motor neuron. Again, an action potential would be an excitatory stimuli that will result in some sort of action at the effector level or at the muscle level. Okay, so this action potential is sent from the alpha motor neuron. When it reaches the end or the axon terminal of the neuron, it will release a neurotransmitter as ACH, which is also short for acetylcholine, into this neuromuscular junction. Again, we said the neuromuscular junction is a place where the neuron and the muscle are going to meet, but they don't actually physically touch. So this axon terminal will release acetylcholine in like this little backpack into this gap between the neuron and the muscle cell. Acetylcholine is going to be running around in this little gap and will bind to receptors on the muscle cell wall. Once acetylcholine binds to the muscle cell wall, the muscle cell has gates that open and sodium comes into the muscle cell. As sodium comes into the muscle cell, this causes the muscle to be depolarized or excited or stimulated, you could say. So once sodium enters into the muscle cell, the muscle becomes excited or depolarized. Okay. So now that the muscle cell has been excited, an action potential will be generated in the muscle potential. Again, now we're ready to send this excitatory message everywhere within the muscle. So we're going to send this signal down the T tubules. Again, T tubules is the same as transverse tubule, and this is like a ditch or an invagination within the muscle cell membrane. So this signals the release of calcium from the muscle's sarcoplasmic reticulum into the sarcoplasm. Again, remember, sarcoplasmic reticulum is a storage bin for calcium. Once this excitatory stimuli has been received at the muscle, we're ready to release calcium into the sarcoplasmic reticulum, into the sarcoplasm, Okay, so once calcium is free to go about its way and move around in the sarcoplasm, it goes over to those protein filaments that we've talked about. Remember, we have the sarcomere set up as actin and myosin kind of overlapping each other. And calcium is going to head over that direction and will bind to this small protein molecule known as troponin. Troponin tells tropomyosin to uncover the actin binding sites. So once calcium is bound to troponin, tropomyosin can now move out of the way. Now we have these binding sites uncovered. As soon as we have these binding sites uncovered, those myosin heads will bind to the actin molecule, and this is what actually is muscle contraction. All right, so when myosin and actin bind to each other, that causes force generation. Okay. So I have a few images on the next page to kind of review these steps, review this sequence as we move forward. Okay. So here you can see the alpha motor neuron. Here is your neuromuscular junction here. Again, notice that they're not actually touching each other. We do have a small gap between the alpha motor neuron and the muscle cell. When the alpha motor neuron has sent this signal and it reaches this axon terminal, also known as the synaptic terminal, it's going to release acetylcholine. Acetylcholine is released into this neuromuscular gap or neuromuscular junction. and it's going to bind to receptors on the muscle cell wall. After the acetylcholine has been bound to the receptors, sodium channels open, sodium rushes in to the muscle cell here. And that causes depolarization or excitation of the muscle cell. This excitatory signal will be sent down the T-tubules, and that will allow us to send the signal very rapidly throughout the entire cell. Once the T-tubules have kind of received this message and this depolarization is spread, it will cause the release of calcium from the sarcoplasmic reticulum, which is conveniently located right next to the T-tubule. Okay, calcium will be released into the sarcoplasm, which is the same as the cytoplasm, and it will come over to actin and bind to troponin. Okay, calcium binds to troponin, tropomyosin moves out of the way, myosin binds to actin binding sites, boom, muscle contraction begins. Okay. We also will continue muscle contraction so long as calcium and ATP are present. Again, remember, in order for this interaction to occur, we have to have ATP, which is our energy currency, in order for us to do this work. So the contraction will continue as long as we are continuing to stimulate the muscle, as long as we still have calcium, and as long as ATP is present. Okay? So in order for us to stop the contraction, we are going to pump calcium back into the sarcoplasmic reticulum so we can store it. Actin covers up those binding sites, so troponin and tropomyosin move back to their resting configuration and cover up those binding sites. Acetylcholine is removed from this neuromuscular junction through an enzyme known as acetylcholinesterase. Okay. As we just mentioned, to stop the contraction, muscle contraction, we have to remove acetylcholine, we have to remove calcium in order for these things to be stopped. So muscle contraction will continue as long as calcium is available. At the end of muscle contraction, calcium is pumped back into the sarcoplasmic reticulum, and troponin and tropomyosin return to their resting configuration. And acetylcholine is removed from the neuromuscular junction through the enzyme activity acetylcholinesterase. And basically that like splits it up. It kind of like digests the acetylcholine. We can reuse part of it, but we don't for some reason we don't get to just like push it back into the neuron. Okay, this is one more image that shows you the same exact process. So let me get my little marker. Here is your alpha motor neuron. Here is your axon terminal. So this action potential or excitatory signal reaches the axon terminal. We release acetylcholine into this neuromuscular junction. It binds to the cell membrane. This causes sodium to rush in to the muscle cell. This causes depolarization, which travels down the T-tubules and all across the surface of the cell, the muscle cell. After the excitatory signal has reached the edges of the T-tubule, calcium is released from the sarcoplasmic reticulum. Calcium has its interaction with actin and myosin. Myosin binds to the actin molecules, and interaction there causes the actual contraction to occur. If we want contraction to stop, calcium is removed and pushed back into or shuttled back into the sarcoplasmic reticulum, and troponin and tropomyosin return to their resting configurations. Here's a link for a YouTube video that essentially shows the same process. It's just an animation, if that would be helpful for you. I would recommend watching it. I know we all learn in different ways, whether it's through seeing or hearing or writing, or whatever method works best for you. I would recommend watching this video that will kind of give you one more view or one more look at how this process occurs. Okay, now we are going to move on to the second part of this lecture, and we're going to be talking about the components of the thin and thick filaments to start. So the thick filaments are composed of myosin is the major backbone in terms of the protein, and this is actually composed of two protein strands that are twisted together, and one end of the strand is folded together to form a globular head. So here you can see in the image above the tail, which is made up of those two protein strands kind of twisted around, and then you've got two globular heads at the end of this protein strand. So these globular heads are going to project from the thick filament and interact with the actin strands, and the number of globular heads that you have is directly associated with how much strength you have the capacity to produce. So the more myosin heads binding to actin, the stronger the contraction is, or the greater the amount of forces that you can produce. The thin filament is actually composed of three types of proteins. So we have actin, troponin, and tropomyosin. Okay. The thin filament also contains active sites to which the myosin heads can actually bind. And these active sites are located on the actin molecule, essentially. So the actin forms the backbone of the filament, and this is made of individual globular actin molecules that are joined together to form a strand. So you can think of this as like a strand of beads, and each individual bead represents one individual actin molecule. Together, again, this will form the backbone of the thin filament. Then we have tropomyosin, and tropomyosin is a tube-shaped protein that twists around the actin. So if you can think about having a strand of beads, this is like a string that would be raveled around the outside of your string of beads. Lastly, we have troponin, and troponin is the smallest of the proteins, and this acts as a binding site for calcium. So this is, like I said, the smallest of the components related to the thin filament, and it's going to be located on the exterior area of the thin filament, and this will allow for a receptive site for calcium to bind. Okay, so here you can see the various proteins. Let me just get my little handy dandy marker. So again, troponin will serve as a binding site for calcium, and again, it's all these just little dots located on the thin filament. So each of these will serve as kind of a binding site for calcium. And when calcium binds to troponin, this is what actually triggers the interaction of actin and myosin to occur, because as calcium binds to the troponin molecules, that will cause tropomyosin to move out of the way, and that will allow the active sites to be uncovered, and therefore actin and myosin have an interaction. So again, tropomyosin's job, this little strand that's kind of swiveled around our beads here, its job is to cover up those active binding sites when the muscle is relaxed. So when we don't want muscle contraction to occur, tropomyosin covers those binding sites and prevents interaction of actin and myosin. Lastly, you can see the actin molecules. Okay, so these are the beads. Again, it's a double strand of beads that are kind of swirled around each other. Each individual globular molecule here represents one individual actin molecule. So we've mentioned calcium, and so you have an idea that it is an important component of muscle contraction. But let's talk about exactly what does calcium do. So once calcium is released from the sarcoplasmic reticulum, it will move into the sarcoplasm. Again, that's like the cytoplasm of the muscle cell, and it's going to bind to that troponin molecule. Once calcium binds to troponin, this will initiate the contraction process, and this occurs by tropomyosin moving out of the way and uncovering those binding sites. So without calcium available and without calcium binding to the troponin, muscle contraction cannot occur because tropomyosin would then be blocking those actin binding sites and therefore preventing us from having an interaction of actin and myosin. So this process, the sliding filament theory, gives us an idea of how muscles actually create movement. So when a muscle contracts, the fibers are going to shorten. And essentially what this has to do with is there's not actually a change in the length of any of the proteins or any of the fibers within the muscle itself. We basically just slide them over the top of each other. So we're just squishing them together, essentially. So if you think about, I'm trying to think of a good example here. If you have a bunch of straws, if we spread them out so that they're not all exactly in a row, that would create, let's say, a 1 foot. We could have like 1 foot of length. And during contraction, we push all of these straws together so that they're all lined up next to each other. And maybe now, instead of being 1 foot, it's only 8 inches long. So we didn't actually change the length of any of those straws, but as we smush them together, the total length becomes shortened. And that's basically what happens with the sliding filament theory, which is how muscles actually shorten. So first thing that happens is we have cross-bridge formation. So when actin binding sites are uncovered, again, as we said, these binding sites are uncovered because calcium binds to troponin, tropomyosin moves out of the way and uncovers the binding sites. So when that has occurred, now myosin head will bind to that actin binding site. Okay, so now they have an interaction. Once myosin binds to the actin molecule, number 2 happens, what's called a power stroke. And this results in a conformational change in which the myosin head tilts and drags the thin filament towards the center of the sarcomere. Okay, then the head is going to pop off or become detached from the thin filament, and this will allow the head to rotate back to its original position. And lastly, we have reactivation of the myosin head, and basically this would allow the myosin head to attach to a new active site. This reactivation process requires an energy input of ATP. And so, if we have ATP available, muscle contraction can occur, and this whole process will continue to occur kind of automatically as long as calcium is present, or until the thin filament runs into each other. And that's when we have kind of a full contraction, when there's nowhere else for those fibers to go. They're butting up next to each other. Okay. In terms of the energy used for this contraction, for each cross bridge that binds to an active site, ATP must also attach to provide energy needed for the muscle contraction. So ATPase is the enzyme needed to break down ATP to ADP and hence allow the contraction to occur. So again, as we said, we need energy in order for this process to occur. And this actually accounts for a large amount of our energy expenditure, is the amount of energy used during this cross-bridge cycling system. So the attachment, power stroke, detachment, and reactivation of the myosin head actually costs us energy, and therefore this accounts for a large amount of energy that we use on a day-to-day basis. The ATPase will actually help us determine how fast this contraction can occur. So one of the major differences between fast twitch and slow twitch muscle fibers is that fast twitch fibers have a very fast acting ATPase, or a very fast acting enzyme, whereas slow twitch fibers have a slower ATPase. So they are slower at completing this cross bridge cycling. Okay, so here we can kind of have a look at what's going on during the cross bridge muscle contraction. Okay, so over here, let's take a look over here. Over here, you can see the thick filament. Here's your myosin head. Here's the actin thin filament. So we are ready to have a contraction. So let's say the binding site has been uncovered. Okay, so first thing that happens, we're going to move this direction. We see cross bridge connection. Okay, so again, as the myosin head binds to the actin, that forms the cross bridge. Okay, now we're moving on down. Here you can see the power stroke occurring, and basically this myosin head is going to ratchet the thin filament and kind of pull it towards the center of the sarcomere. Okay, so the myosin head kind of pulls the actin molecule forward. Okay, so then after power stroke has occurred, we're going to have a detachment of the actin molecule. Sorry, I don't know why this blue is already on here, but we're going to have the detachment of the myosin head. So the myosin breaks away from the actin thin filament. And then we're going to have the reactivation, which requires ATP molecule. Okay. Again, if you look here on the left, we can see how the sarcomere actually shortens. Okay, so let's look here at this image. So this was my example of the straws that are not all in line. They're kind of spread out. So you can see actin thin molecules here and the myosin in the middle. And during a relaxed state, you can see that they're somewhat overlapping, but not a lot. We've got some room to move. If you look over this way, we've got room to move. So during contraction, we are literally going to pull the actin. Let's get a different color here. We're going to pull the actin molecules towards the center. Okay. So those myosin heads, as they attach and pull, they're going to pull the actin towards the middle of the sarcomere from both ends. Okay, so you can see with the blue arrows here, actin is being pulled towards the center. During a contracted phase, you can see that everything is butted up next to each other. So when the contraction is fully completed, or we get to like a full contraction, those actin molecules don't have anywhere else to go because they're butting up next to each other here in the center of the sarcomere. And like I said, that would be considered a full contraction. So just to kind of reiterate, keep in mind that the length of the muscle fibers themselves do not actually change, but the overlap of each other is going to allow them to slide inward in order for this muscle contraction to occur. So again, those thin filaments are going to be sliding towards the center of the sarcomere, and as they slide towards the center of the sarcomere, this is what actually allows the muscle to shorten and therefore have its contraction. Okay, here's a link for a YouTube video here at the bottom. I would highly recommend it. This just kind of gives you another visual. It's going to show you exactly how this muscle contraction, how the sliding filament theory actually works. So please take a second here to stop the PowerPoint and have a look at this video that will actually demonstrate how the cross bridge, those 4 steps, occur, and it will kind of review those steps for you. Okay, so what happens when we want to end the muscle contraction? Again, we need to get calcium out of there because we know that calcium is what allows those binding sites to be uncovered. So if we can remove calcium from the situation, this will allow troponin and tropomyosin to return to their resting conformation and cover those cross-bridge binding sites, therefore preventing actin and myosin from having an interaction. And basically how this happens is calcium will be pumped back to the sarcoplasmic reticulum, which again is its storage bin or its place to keep stored. Okay. So we've spent some time talking about the sliding filament theory, how we can allow muscles to shorten. But it turns out we actually have 3 different types of contractions. So far we've talked about what's called concentric contractions. Okay. In most sports and exercise and really in any time of your day or life, we're using all 3 types of these contractions in order for us to execute smooth coordinated movements. So concentric contraction are the shortening of the muscle. So again, we're talking about sliding those fibers in towards the middle of the sarcomere. Okay. So concentric contraction is shortening. Isometric contraction, iso means the same, metric means length. So with isometric contraction, the length stays the same. So we're generating force with no change in muscle length. That's isometric contraction. And eccentric contraction is actually the lengthening of a muscle and also producing force. So there's a difference between just relaxing your muscle and eccentrically contracting the muscle. So during an eccentric contraction, we have to be under control and producing force against gravity or against some sort of weight in order for it to be considered eccentric. So with concentric, we're shortening, so therefore the muscle fibers are pulling towards the center of the sarcomere. With isometric contraction, we're generating force, but we're not shortening or lengthening the muscle. So a plank would be an example of this. We also are isometrically contracting a lot of postural muscles while you're just sitting here. And then eccentric contraction, again, we're lengthening the muscle fibers. So those fibers are kind of being pulled apart from each other in a controlled way and therefore generating force while lengthening the muscle. So how much force do we need? So the amount of force produced or developed is dependent on several factors. So again, what determines how much force we need to use in a given situation? Well, the first thing that happens is we're going to use our past experience. So for example, if I see a book sitting on a table, I can use memories or past experience, past situations, and take a look and see if, I can remember how much it was to lift a book, the last time I did it. So I'm going to use that to kind of judge how much force I need to produce, because we're not going to generate a full maximal contraction every time we have any contraction occurring. So we can use kind of our past history to kind of get a ballpark in terms of how much force we should actually produce. And that will allow us to make some decisions about the following factors. So the amount of force developed is dependent on the number and type of motor units stimulated, the frequency of the motor unit stimulation, the size of the muscle fiber and sarcomere length, and muscle speed of contraction. Okay, so let's kind of run through these here a little bit more in detail. So the first one we said was the number and type of motor units stimulated. So the more motor units I stimulate, the greater the force of contraction can be. So in order for me to lift up that book, let's say it's a pretty small book, maybe I need to activate 3 motor units in my bicep muscle to do that. Okay. Let's say I decide I need to lift up my dog, who is like 80 pounds. So now, in order to do this, maybe I'm going to activate 6 motor units. Again, the more motor units I activate, the greater the amount of force will be. Also, the type of motor units is important. So, which do you think would generate more force? Fast twitch fibers or slow twitch fibers? For those of you that took the AMP1 class, you should know that type 2 fibers or our fast twitch fibers should produce more force for us in comparison to our slow twitch. Also, the frequency of stimulation. So again, the more frequently I stimulate the muscle, the greater the amount of force will be. The size of the muscle, again, a larger muscle will have the capacity to generate more force in comparison to a small muscle. Again, as we already mentioned, muscle fiber type. So again, fast twitch fibers would allow us to generate more force compared to slow twitch. Also, muscle fiber and sarcomere length as well. So we have an optimal length tension relationship. And with this length tension relationship, it can allow us to determine the greatest capacity for strength is kind of like in the middle of your range of motion. So again, if you think about, and again, a lot of you have kind of already heard about this, but if you think about doing a bicep curl or something, the hardest part is the beginning of the action. So when your arm is completely extended and you're trying to get the weight started, that's kind of the hardest part. Once you get to that middle of the range of motion, we have an optimal overlap of actin and myosin molecules, and so therefore you can generate the greatest amount of force in that place. As you move to your fully contracted position, those fibers are going to run into each other, and so therefore you decrease the ability to generate a high amount of force. Also, the speed of the muscle contraction will make a difference. So the greater the speed, the less force you can produce, and we'll talk about why that is here coming up. Okay, so this is looking at that speed of contraction issue. Okay, so this is the force velocity curve of a muscle. So let me get a different color here. So let's start by thinking about concentric contraction. So we're over here on this side of the graph. With a concentric contraction, if I have a very light weight with a bicep curl, let's say you give me 5 pounds. With 5 pounds, I can whip that weight around really quickly. So I can generate force at a very fast velocity. I can move it at a very fast speed. Okay, 5 pounds is super easy. Let's say you give me 10 pounds. At 10 pounds, that's a little harder. That's kind of hard for me. So in order for me to generate the force, I actually need to slow down. So at 10 pounds, I can do the contraction. It's a lot harder. So I actually slow down in order for me to have the capacity to lift that weight. Okay. Then let's have a look at isometric contraction. So let's say you hand me a 20-pound weight. That's really heavy for me. So with a 20-pound weight, I can't generate enough force that I can shorten the contraction or shorten the muscle, but I can produce enough force that I can hold it in an isometric position. So at 20 pounds, the force is so great that I can hold it isometrically, but I'm not strong enough to lift it in a shortening way. Okay, so again, as we're increasing the weight, you can see that the velocity is decreasing. Okay. Then let's say for some crazy reason you hand me a 30-pound weight. That's way too heavy for me. But you could hand it to me at the top of the contraction and I could lower it eccentrically. I could lengthen the muscle and therefore have a lengthening contraction under some amount of control with 30 pounds. Okay, it's way too heavy for me to have a concentric contraction. It's even too heavy for me to hold it isometrically, but I can allow myself to lengthen the muscle in a controlled manner with this. Okay, so this just kind of shows you the same exact thing. We're just looking at it in a different way. Okay, so as the speed of the contraction increases, you can see that the resistance applied must decrease, and vice versa. So the heavier the weight is that you're handing me, so If you hand me a very heavy weight, I can move it very slowly. If you hand me very little weight, I can move it very quickly. Right? So there's an inverse relationship between speed and resistance. So if speed increases, resistance decreases. And vice versa. As resistance increases, speed must decrease. Okay, so they're inversely related. Okay, so let's talk about this length-tension relationship that we've kind of alluded to. So the length-tension relationship is basically defined as an optimal overlap of the actin and myosin filaments, which allows us to maximize cross-bridge interaction. Okay, so the length-tension relationship has to do with how much tension can be generated based on the length of the muscle. So as we said, there's an optimal level of overlap associated with actin and myosin filaments that will allow us to maximize force production. And again, that's generally going to occur in the middle of our range of motion. Okay, so here is an excellent example, and again, you either have or you will get the opportunity to actually measure or experiment with the length-tension relationship in our labs this course. So let's have a look-see here. Let's look at this end over here. Okay, so here we're looking at the sarcomere length is very stretched. So when you're in a fully stretched out position, there's not a lot of cross-bridge interaction with the actin molecules. So those little blue lines represent each cross-bridge, and we want them to be close enough to the actin that they can have a very easy interaction. And if they're not close enough, then therefore they're not available to produce force. And that's what's happening when we're in this extended position. Okay, so there's not enough cross bridges available to interact with the actin, and so therefore we have a decrease in our force capacity. When we get to the middle of our range of motion, this is the optimal level of overlap. And again, now you can see these little heads sticking out, and they are available to have an interaction with the actin molecule. So that's awesome. That's what allows us to produce the greatest amount of force possible, is if these myosin heads are all available to interact with the actin molecule. So that's why we can produce the greatest amount of force in the middle of our range of motion. Now we continue to shorten and shorten and shorten, and finally we get to a completely contracted position and there's too much overlap. So again, sure, those myosin heads are available to have an interaction with actin, but everybody's already butted up next to each other in the middle. So therefore, we can't have any more interaction. We can't produce any more force because of the relationship of actin kind of running into itself. Okay, so now we're going to switch gears a little bit and talk briefly about muscle fiber types. So all muscles contain a combination of single skeletal muscles that have differing shortened speeds and strength capacities. So all of us have type 1, all of us have type 2A, and all of us have type 2X fibers kind of intermingled throughout the entire muscle. Okay, so we all have all three types. The amount that we have of each type can vary from person to person, and we'll talk about kind of, what that is related to and that sort of thing here coming up. So sure, we all have all these types of motor units or all these different types of muscle fibers, but one single motor unit is made up of one single type of muscle fibers. So if you think of one single motor unit as a factory, each factory has one type of workers. So you either work in a fast factory, or you work in a slow factory, or you work in an intermediate factory. Okay, there's not a combination of slow and fast people within a given factory. You're either fast in the factory or you're in a different factory that's slow. So everyone in the factory is the same type of person, or everyone in that given factory is the same type of muscle fibers. Okay. So, again, a motor unit basically represents our factory that we just described, and an alpha motor neuron is kind of like the boss, and the muscle fibers that he innervates are all your workers in your factory. So we can have a large motor unit, and a large motor unit is made up of greater than 300 muscle cells and is usually a fast fiber. Okay, so usually fast fiber, type 2 fiber, type 2A, type 2X fibers are going to be large motor units. So again, you can think of this as like Domino's. So let's think about it this way. Domino's has a lot of factories and a lot of people work in their factories. And they're very fast. The reason that they're so fast is because they've been able to perfect the art of making pizza in a matter-of-fact way. Maybe you could think of it that way. I don't know if any of that's actually true, but anyway, we're going to go with it. So that's like your large motor unit. They're more efficient, and that's why they're faster. Okay. A small motor unit, you can think of that as like El May downtown. It's a small restaurant, and there's not a lot of people that work there. So it might take a lot longer to get a pizza there because there's not as many workers there. Okay. So a small motor unit, or in this conversation, El May, is made of less than 300 muscle cells, and they're usually slow in nature. So type 1 fibers. And again, these type 1 fibers are going to allow for fine-tuned movements, small, minute, delicate types of movements that maybe don't require a large amount of power and don't require a large amount of force. Okay. So muscle fiber recruitment occurs when an alpha motor unit is activated. All the fibers within that unit contract. So you are either everyone in the factory is working or no one in the factory is working. Nobody gets half the people come in, half the people don't. You're either all working at your factory or nobody's working. The factory is completely shut down. Okay. So in order to generate more force, we can activate more motor units. So we can have more factories go to work in order for us to produce more force or to make more pizzas or whatever we're doing. So initially we want to activate type 1 fibers first. So we're going to activate those slow twitch fibers first, followed by type 2A, and then lastly type 2X. So slow twitch are activated first, type 2 are activated second. So that's the order in which muscle fibers are going to be recruited. Here you can kind of see that interaction. So we've got, motor unit 1 recruited. Again, if you look up here, these little blue guys represent motor unit 1. So motor unit 1 is a slow twitch fibers, or, your slower moving fibers, your El May types of factories. So they start making some pizza, but again, they're not very fast. They can't produce a lot of pizza very quickly. So if we need more pizza, we're going to activate another motor unit. Okay. And this motor unit is yellow. You can see here, the yellow ones are a little bit bigger. So these are like a medium size factory, and they're going to be a little bit faster. So now they're starting to really bang out pizzas. We're producing more force here. Okay. And then lastly, we're going to activate our motor unit 3, or our fast twitch fibers. And that's going to allow us to produce a really great amount of force, okay, and very quickly. So that's when you call in Domino's, because we really need a lot of pizza really quickly. Okay. So type 1 is first activated. That produces a low amount of force. It's slow in nature. Then we activate type 2, which is kind of like an intermediate type of fiber that's kind of fast, but not as fast. And then lastly, we'll activate our fastest types of fibers, and that will allow us to produce the greatest amount of force. Okay. So in terms of types of muscular force, we have a couple different terms that we want to kind of discuss. So muscle strength has to do with the greatest amount of tension or the greatest amount of force that you can produce at any given time, whereas muscular endurance has to do with being able to move a submaximal intensity multiple times. So we could think of muscular strength as your max out on bench press. How much weight can you move one time? So we're looking at, what's the greatest amount of force that you can produce. Whereas with muscular endurance, we want to look at multiple repetitions at a submaximal intensity. So maybe you could think of that as the maximum number of push-ups you could do without stopping. That would be a test of muscular endurance. And then lastly, muscular power has to do with both strength and time. So we're not only concerned with the amount of force that you're generating, but we also want to know how quickly you can do this. Okay. I would say a majority of sports actually use all three types of muscular force within the given parameters of their activity. So in terms of fatigue, what actually is fatigue? So with fatigue, we're looking at a decreased ability to generate force. Okay. And again, this could be related to a lot of different things. It could be related to, something that's going on within the muscle. It could actually be related to psychological fatigue and other things like that. So there's a lot of reasons that we have fatigue, and you'll actually get the opportunity to talk about fatigue in your lab reports after we've done the clothespin experiment, where you actually get to measure muscular fatigue in your fingers while you're doing this experiment. So in terms of causes of fatigue with muscular strength, so again, if you're thinking about the ability to generate the greatest amount of force possible, fatigue could be caused by a variety of things. Maybe, it's a neurological fatigue. So you can't produce any more force because you're limited neurologically. Could be a hormonal issue. Could be a mental or psychological issue. Could be related to the buildup of waste products as well. So there's a lot of things that kind of limit us in terms of fatigue. With muscular strength, though, it might be likely that, maybe you don't even feel fatigued. Maybe that's not a limiting factor for you. Maybe it's just you don't have enough muscle mass to generate a greater amount of force. So there could be a variety of things causing fatigue for strength. For muscular power, kind of the same type of thing. Maybe you're limited by the type of muscle fibers that you have. So if you have a large amount of slow twitch fibers, you don't have a great capacity to generate speed, most definitely. Again, neurological factors can be certainly related to muscular power. Waste products associated with metabolism of, fast types of movements would certainly be a possible cause of fatigue. So those are definitely a possibility. Maybe we run out of fuel. That could also be a possibility with muscular power, again, depending on how long the exercise bout is. Maybe we run out of fuel available as well. Maybe you're just limited by the size and training type of your muscle fibers as well. And with muscular endurance, I would certainly say that psychological fatigue is a huge issue with endurance types of things, most certainly. Fuel, again, maybe we've depleted our fuel sources. Maybe we have a greater amount of waste buildup. Maybe that's your limiting factor. So there's certainly, several possibilities with endurance. But most likely, I would say it's a buildup of waste products, which decreases our capacity to generate force, or psychological fatigue. And again, you'll have the opportunity to talk about, either or both of those factors in your lab report.